Well, thank you for inviting me. I was debating whether to do the talk in English or in French for all my Francophone friends here, but I'll stick with English for this morning. So yes, I've been with USDA at the Natural Products for the last 20 years. And my work, as Linda mentioned, worked on the mode of action of natural products, but about a half of my work has also been involved with the mode of action of synthetic compounds. And I've been asked with my boss, Steve Duke, to write a review that Linda mentioned. And I'll just mention that most of the talk will be based on that review. All of the slides, many of those slides are available at the plant physiology website for your lectures if you're interested. And I also have a PDF of the paper, or if you want the slides, I don't mind sharing at all. So I've been at the USDA for 20 years, but I thought it was time for a change. And so actually, starting in January, I'm leaving the USDA and I'll be at Colorado State University working with Todd Gaines and Phil Westra and Scott Nissen and others. And my work will then flip back into the mode of action and mechanism of resistance to herbicides. I also want to say that I'll be looking for 2 or 3 outstanding graduate students. So I'll put a plug here that if you're interested in the mode of action and herbicide resistance, maybe come talk to me afterwards. All right, so this is the review that Linda mentioned. We published this in 2014 in Plant Physiology in a special issue that focused on weed control. And the idea was, what are the potential of using natural products to control weeds? So a little bit of background. Let's start with this. About 80% of all the drugs that are commercialized for human health are either natural product or derived from natural products. So it's a huge impact of natural product on health. Actually, about 11% of all sales in agriculture are derived or natural products as well. So it's quite a bit of an impact. And this has been particularly useful in the area of disease control and insect control. The impact of natural products on the development of herbicide has not been as good. And I'll show you some data that we've compiled in another paper that was published in the Journal of Natural Products, where we looked at all the EPA-approved registration from 1997 to 2012. That's at the time when we did the paper. And what we did is we looked at the number of active ingredients and which ones were natural products or not. And it's kind of hard to see from here, but ultimately, most herbicides, of course, will be synthetics. But there's about 20% of all pesticides in that time period from 1997 to 2012 that were either natural products or compounds that were derived from natural products or biologicals. So that's, 20% is not too bad. Now, if you look at herbicides, though, it's not quite as important. And again, you have to remember the dates that I'm looking at, is this time period. So that 8% of the impact of natural product on the discovery of herbicides really only talks about the trachetones, because the compound like glufosinate was discovered before this time period. So the impact overall is greater, but recently it has only impacted about 8% of the herbicides. So I'm transitioning now to maybe how chemists in the real world have done discovery and how maybe nature has done discovery. So one of the popular approaches, high throughput screening target-based. And essentially, you start with a large number of compounds, 100,000, 200,000, 300,000 compounds. And you screen toward a particular target site of interest that biologists have identified as a potential target site for herbicide. And it's an iterative process where you do a high throughput screen, you end up with many hits. Then you do compound selection, you have fewer hits, and very often with a validation from enzyme assays, you end up with 1 or 2 lead compounds. And at that time, then it's a very short time, very large amount of compounds, and then chemists do some elaboration on the structure that's discovered to maybe develop something that's more appropriate for commercial use in the field, and with some kind of a structure-activity relationship. And at the end, you end up with an optimized compound. Another approach has been what's called biorational, where you define a protein of interest, and then you can identify the binding regions. They, using computer modeling, try to find a framework that would fit in that binding pocket. Then you optimize, and in the end, you end up with an active compound. All right, so this is basically how things have come to be to date. How does nature do it? And nature is a little bit different. Of course, it doesn't do things very quickly, but it has time on its side, right? It does things over a long time. But the advantage is it can look at many, many target sites, many, many different classes of compound at the same time over a long time. So the high throughput is not how quickly it gets down to the target, but really the vast amount of interactions between chemistry and biology that's going on over time. And so, for example, you have many chemical interactions happening over a long period of time. And then some of them will be positive, like mutualistic or symbiotic. And these are good interactions. Or over time, you find some more negative interactions, like competitive interactions and parasitic interactions. And biology, over long periods of time, develops novel pathways, very often through gene duplication and mutations, and that takes a lot of time and selection. But in the end, plants develop secondary metabolites that either enhance the positive response by producing compounds that would attract, let's say, the butterfly to you, to your own benefit, or producing compounds that will be toxic to the parasite. In the end, though, that high throughput, but not over a short time, but over a very long time, leads to a competitive advantage. And that really involves very complex sets of interactions. And when we talk about natural products, I'll show you the end product, but these end products are the results of these long processes of interactions. Okay, so some of the work that I've done in Oxford, Mississippi with many chemist colleagues is you start with instead of a pure compound, you start with something like this, a kind of an extract of a plant or a fungus or some other organism. You do some extraction, some chromatography, and then you end up with some pure compounds. You ultimately identify the structure of the compound with NMR, and in this case, I'm showing you sorgoleon, a compound that I've worked for many years on. And so this seems very straightforward, but if you work with natural product chemists, it's not as easy as it looks, right? It takes a lot of knowledge, and technology nowadays makes that easier because of the accuracy and the amount that are required for characterization are much less than it used to be. So this is a little bit of how we got to where we are. So now I want to step back and talk to you about plants in general and what we know about herbicide mode of action, and how natural products have been selected through the evolution to target these very specific sites. So what do we know about photosynthesis, for example? Photosynthesis is a very integrated process that involves many pathways in plants. And when, very often you think of photosynthesis, you think of chlorophyll, electron transport, and maybe carbon fixation. But actually, in plants, it's such a core function that there's many integrated relationship. So for example, in photosynthesis, you have, and where's my pointer here? Photosystem 2, and there are herbicides that inhibit the flow of electron in photosystem 2. And that affects the flow of electron. That also is involved then in photosystem 1. So photosystem 2 and photosystem 1 are quite related. And they are also inhibitors of that pathway. But it's more complicated than this because to have photosynthesis, you need to have chlorophyll. And so there's a whole pathway leading to chlorophyll synthesis. And if you start disrupting this, then you will also very likely hit target sites that would be good for herbicides. And indeed, there are some. I'll talk about that in a minute. But then you also need carotenoids for photosynthesis. Without carotenoids, your plant dies because the chlorophyll is destabilized. And so there are, this is the whole pathway actually to carotenoid synthesis. And there are inhibitors in this area, there are inhibitors in this area. But it's even more integrated than this. For the electron transport, we need plastoquinone. And it turns out that in this pathway, you have the HPP pathway that leads to plastoquinone synthesis. And so there are inhibitors here that affect photosynthesis. But also, PQ is an essential cofactor for phytoin desaturase. So if you block this pathway, you also inhibit PDS. So this is just to show you that through evolutionary processes, organisms do things differently than we do. We tend to focus on one target site and try to find inhibitors of a specific target sites. Through evolution, natural products have looked at the composite, for example, of photosynthesis, and what are the potential parts of the pathways that can overall have a negative effect on photosynthesis, but not only looking on one specific step. So for example, then what we know as far as natural products, if you look at the Z scheme right here, typical thylakoid membrane, you have your PS2, your PS1, your TPAs, and all of this is involved in the light reaction of photosynthesis. Well, are there natural inhibitors to these pathways? Well, I'll talk to you more about this in a minute, but of course, sorgoleon looks like plastoquinone, and it competes with the binding site of plastoquinone on the PS2 right here. But there's more examples. I can't even read. Aractocin C inhibits the water splitting complex. Okay, this is an interesting one right here. This is a compound that was discovered by Dow Agro. That's actually it has a positive charge, it's a cation structure, and it actually competes for the electron transport in the same way as paraquat does. It's an electron diverter. This one affects the proton gradient in the membrane, and that last compound right here, tentoxin, inhibits the ATPase. So this is just an example of how natural products have looked at photosynthesis as a key process for plant survival, and has, through evolution, developed a series of compounds that can affect plant photosynthesis by looking at basically every aspect of the processes involved. So I'm going to tell you a little bit more about sorgoleon, which is a compound that I've worked on for many years. So sorgoleon is interesting because you can grow sorghum bicolor in the field, kill it, and then plant soybean directly in it. And what you can see is you have very few weeds growing in it. And what we know about this is that sorghum produces a compound that comes out of the roots. So this is a root. You have the root hair. That's a enhanced view of that root hair. And you see that droplet coming out of this. It's full of a compound called sorgoleon. And it has very interesting things. So I think, can somebody click on that image right here? So we wanted to know the composition of that droplet. And so I had a technician use a SPME, and with a macro manipulator under the microscope. Can you do that again? She did that about 200 times and collected 200 droplets, and that was enough to put on the GCMS and analyze exactly the composition of what was coming out of that droplet. And what comes out of that droplet is sorgoleon, this lipid benzoquinone. Okay, and what does that do? That's a very strong photosynthetic inhibitor. It inhibits oxygen evolution by competing for the binding site of plastoquinone. So it acts just like atrazine and diuron. And you can actually displace atrazine with sorgoleon if you bind atrazine to the binding site. What's interesting is it only inhibits photosynthesis in very young plants. This is what we show here. When we tested the efficacy of sorgoleon and atrazine on plants based on plant age, you can see that basically atrazine inhibited electron transport regardless of the age of the plant. But with sorgoleon, it only worked in very, very young plant. As soon as the first leaf came out, it had actually no effect. And that has to do maybe with the way it works in the field, but I don't have time to speak too much about this. What we wanted to know is the plant making sorgoleon at one time and coming out of the root hair, or does it make it continuously? Is it a dynamic process? And so what we were able to do is we took roots, washed them carefully without destroying the root hair, and then we ended up with root hair without any sorgoleon. And then we waited over time to see what happened to the production. And this is what happens. You can see that you can remove all the sorgoleon, but within 1 week, basically the plant made some more. So then what you have to imagine in the field, you have your sorghum, your roots are in the ground, the root hair produce sorgoleon, these get diffused in the soil, and the plant keeps making it. So in a way, it's like a slow-release herbicide. It doesn't make a whole lot, but it makes it constantly over the growth season. We wanted to know how the plant made this, and there's a reason for that is we wanted to know if we could then manipulate the pathway to make sorgoleon, make sorghum produce more sorgoleon, or even transfer the pathway to another crop. So then you would have a crop that would make its own herbicide in the soil. So to do that, we needed to understand the pathway, and we used a method called retrobiosynthetic NMR analysis, where we use some of the substrates like acetate or methionine or sugar, and we wanted to know how it ended up in the sorgoleon. And I'm going to go through that pretty quickly, but essentially, when we use methionine with a methyl group labeled with a C13 carbon, what we ended up with is a carbon labeled only here. So we knew that this carbon came from methionine, and that enabled us to look at that part of the pathway where this carbon ended up here, here, and here. So then you can go back and you can figure out, well, you need an O-methyltransferase to do this. We did the same thing with 2 labeled acetate. And when we did this, we ended up with 3 carbons only here, labeled. So by going backward, looking at the retrobiosynthetic part of this, what we were able to figure out is that to get this, you needed this, you needed this, you needed this. And ultimately, that led us to the confirmation that we needed a polyketase synthase to make this part of the molecule. Then we labeled with 1 acetate, which is this green carbon. And when we did that, we got all of the carbons labeled. So then, using our biochemist hats, we go back and we figured out to do this, you needed this part of the pathway. All right, so then in the end, after 7 years of worth of work, we knew that we needed an O-methyltransferase, we needed a PKS, we needed a fat-2 desaturase, and then of course fatty acid synthase right here, and probably a P450. Well, what we have now, and I don't know if it's my next slide, I better stay here. We now have all the genes characterized. What we know is all the genes are only expressed in the root hair. And we have all these genes characterized. We have some RNAi knockout of PKS. So we have sorghum plants that don't make any sorgoleon because we knocked out this step right here. And what's interesting, for example, is this intermediate right here is present in rice, right? So essentially, one of the things that we're playing with right now is could we make rice more allelopathic? And to do that, all we would have to do is basically clone this gene and this gene to get sorgoleon made out of rice, right? But we're quite a ways from that still. I think, yeah, this is what I just said. We have all those genes isolated, characterized, and now we also have a construct that we can infuse, for example, in tobacco leaves, and then the tobacco leaf dies where it's infused because the tobacco leaf is starting to make sorgoleon, and that, of course, inhibits its own synthesis. So we're making some progress on that. All right. So this is one example of directly the inhibition of photosynthesis. I mentioned about the inhibitors of pigments. If you inhibit carotenoids, you affect photosynthesis. If you inhibit chlorophyll synthesis, of course, you're going to affect photosynthesis. And so these are the different target sites that are known to be inhibited by natural product. This first step, one of the first steps in chlorophyll synthesis, is inhibited by gabaculin. Safran inhibits that site, which is the target site of PPO's inhibitors. Phosmidomycin inhibits the second step in the non-mevalonic pathway. And the first step will be the one inhibited by chromosome, right? But nature selected this one right here. Leptospermone, which is the backbone that was used to derive the trachytone herbicides, inhibits HPPD right here. And I'm showing this one because there's some evidence that synmethylene, which is not a synthetic, it's not a natural product, but this component is a natural product right here, the 1,4-cineol, may actually inhibit the first step in tyrosine aminotransferase right here. All right, so then for this one, I'll focus a little bit on leptospermone. I've got some work on this, but there's a lot of literature that shows that these herbicides were derived from leptospermone. And it was discovered by noticing that there was very few plants growing around this plant right here. And they took soil samples and chemists isolated leptospermone. And in the end, with structure optimization, they ended up with new trachytone herbicides. And so we worked with leptospermone with a plant from New Zealand called Manuka. And Manuka has an oil that has a lot of antifungal and antimicrobial activity, and it's rich in those trachytone compounds. And when we did some analysis, we discovered there was these 4 trachytones. Which one is this? This would be leptospermone, which was the most abundant one. And where am I going? And this is produced in very specialized cells inside the leaf structure. They are called asagenous cells, sagogenous cells. And that's for protection because leptospermone is a herbicide. So a plant that's making a herbicide has to be protected against its own herbicide. And it does that by compartmentalizing it in a very specialized structure so that it's not affecting the cells around it. And as opposed to fat-2 desaturase inhibitors that inhibit that step right here, like this compound noflurazone, inhibit fat-2 desaturase. That, of course, leads to carotenoid inhibition. What we have now is nature selected a different target site that leads with the same results, bleaching of tissues. But the way nature did that is by synthesizing a compound that actually, like leptospermone, that affected this herbicide right here. Sorry. So by inhibiting this target site, you inhibit plastoquinone synthesis, and plastoquinone synthesis is required for fat-2 desaturase, so that in the end, you have no carotenoids, and you end up with bleached plants. So you have a very similar phenotype, but the way you get to it is very different. Nature has kind of selected a very longer road to the same outcome. We did some structure activity relationship, and I don't want to spend too much on this, but essentially what we've discovered, this is a catalytic domain that's an iron right here, and this diketo group is stabilized around this iron, and their pocket goes that way right here. And what we've done with natural products is we've discovered that the pocket is very lipophilic, so that the longer the tail, the longer this side chain, the more active the compounds are. We've done a lot of structure activity relationship, and I don't know if I have actually the next slide. Is it going? I think I'm not moving. Yeah, I don't think my slide's moving forward. Here you go. All right, so talked about photosynthesis. You can inhibit the electron transport. You can inhibit the pigment synthesis. What else is important in plants? Well, or in any living organisms, but because we're working with weeds, we're talking about plants. So biopolymer synthesis are important target sites as well. You can either inhibit the building blocks, or you can inhibit the assembly of these building blocks. So what are these examples? You can have inhibitors of sugar synthesis, like some callose synthase inhibitors, or you can have inhibitors of cell wall synthesis. That would be good target sites for herbicides. What about fatty acids? There are inhibitors of fatty acid synthesis, like ACCAs and elongases, or you can have also inhibitors of membranes and cuticles. The assembly of the fatty acid into larger biopolymer. Last but not least, what about amino acids? We know there's target sites like EPSPS for the aromatic amino acid synthesis, ALS for the branched chain amino acids, glutamine synthase for glutamine synthesis. And so you have inhibitors of the building blocks, or you can have inhibitors of the assemblies of those building blocks into macromolecules, in that case, cytoskeleton. And so how, these, we know from our experience that these would be good target sites for herbicides. Well, has nature identified these as good target sites? Well, if you look at amino acid synthesis, phosphorinothricine, which is, I guess, the natural form of glyphosate, of course, inhibit glutamine synthesis right here. That's a natural product. It's the only herbicide of its class, this only chemistry that is commercialized to inhibit this target site. What do we have? Phaseolotoxin inhibits this enzyme right here that is in the pathway between glutamate and arginine. There's no herbicides that have been developed for that yet. 5-methyltryptophan is an inhibitor of tryptophan synthesis, and that's produced by fungi. These were examples of pathways that are important for plant survival, either the synthesis of building blocks of macromolecules or the synthesis of their assembly of these building blocks into macromolecules. And so I gave you some examples on that larger figure, but now I'm going to focus a little bit on phosphorinothricine because that is probably the one success story on natural product as herbicides. Of course, we have mentioned the trachitone herbicides, but these are semi-synthetic. I mean, they're natural product-like compounds. But phosphorinothricine is actually a natural product, and that's the only natural product that is commercialized as a herbicide. You want to press enter? Wait, not yet. So basically, phosphorinothricine, well, it's a non-selective herbicide and inhibits glutamine synthesis. I'm so handicapped with this remote. I wish I could control it from here. Go ahead, press enter. All right, so one thing that's really great about phosphorinothricine is that you can easily detect its effect by monitoring ammonia accumulation. Glutamine synthetase is involved in nitrogen ammonia recycling. So when you inhibit glutamine synthesis, you get a very high accumulation of ammonia, which is what you see here. As you increase glyphosate concentration, you get a huge increase in ammonia. You want next slide? Oh, it's working? Well, maybe I need to be on that side. Let's try that. Are you pressing the right button? Yeah, the green one right here, the big fat one right here. Yeah. And there's, and the enzyme assay is not very hard to measure, which is very convenient. Oh yeah, look at this. So you can measure the inhibition of glutamine synthetase by looking at what's happening at the ATP level. Or you can do it by inhibiting the secondary reaction. And this is colorimetric, so you can easily detect this by spectrophotometry. All right, so how is phosphorinothricine used in agriculture? It's a little bit different. Okay, and chemistry can be very boring, but the key thing is you see this carbon right here. There's a carbon right here, and there's 3 bonds, right? And that bond, that one that goes down to the NH2, can be down or it can be up. And these are 2 different molecules. Well, nature only makes the L form. Nature only makes this form right here. But for a chemist, it's really difficult to make one versus the other. So the way they commercialize it is they make the mixture that has both the L and the D right here. But only the L is active. So basically, when you spray glufosinate, if you spray a pound of glufosinate, you're really spraying half a pound of the active compound. The other half pound doesn't do anything, right? Which is not very, from a synthesis perspective, it's not very efficient, but it's cheaper to do it that way than to purify the L. And what's interesting is not only it's a natural product that inhibits a unique target site, but there are microorganisms that can metabolize glufosinate to make it not active. And so there are plants like the Liberty Link plants that have been transformed to express an enzyme called right here, the phosphoinositrazine acetyltransferase. And they put that enzyme or the gene for that enzyme in the plant. And when you do that, you make plants resistant to the application of glufosinate. And that's really, it's a very simple reaction that it does. If you have phosphoinositrazine, this enzyme, phosphoinositrazine acetyltransferase, uses acetyl-CoA and puts this like CH3O gets attached right here. So it attaches that acetyl group right here to that nitrogen. You end up with N-acetylphosphoinositrazine, and this is not toxic to the plant anymore. So that you can spray your glufosinate, kill all the other plants, but your transgenic crop will not die because you'll take the phosphoinositrazine and very rapidly convert it to this. And we have a paper that we submitted where we actually looked at cotton, conventional versus transgenic cotton, and we showed that within a few hours, all of the glufosinate is metabolized. So it's very, very rapid. Maybe this way? Okay, you're my witness, right? I'm pressing the right one. Okay, anyway, so I'm going to try to skip here because I'm dragging here. Okay, press enter again. I don't know. Maybe I have a magnetic vibe or something. It's, oh, yeah. I don't know what I'm doing. Is this another one? Okay, thank you. All right. So yeah, there are different resistant genes, and I don't think I want to spend too much time on this, but essentially, one thing that we were interested in is some farmers use insect-resistant cotton as a glufosinate-tolerant crop, which is very risky because what people have done is to make a crop resistant to glufosinate, they put the gene that makes it resistant and have multiple copies of that gene. So there's a lot of that enzyme present in the plant. But some cotton varieties have made tolerant to insects tolerant by putting the cry gene. But the way they select it for resistant is they put one copy of the glufosinate resistance, right? So that the cotton that's insect resistant carries one copy of the enzyme that makes the plant resistant to the herbicide. And some farmers try to save money, so they buy the insect resistant cotton. They have good control of the insects, but they also spray glufosinate because the plant has some level of resistance. But that's kind of risky because this is the level of expression of the enzyme of the gene in the one that's made to be herbicide resistant, and this is the level in the one that's made to be insect resistant. So you don't have the same number of copies, and I think farmers can run into problems by, it's like a Russian roulette. It will work once in a while, but you may get to a place where you end up killing your crop, and this is what we're showing here. All right, so we talked about photosynthesis, we talked about pigments, we talked about macromolecules as amino acids. Now let's take a look at maybe membrane function. This is one of the favorite targets of natural products. There's a lot of natural products that target membranes. Juglone is an ATPase inhibitor. This one inhibits NADH oxidase. Syringomycin is a really interesting one. It's produced by a fungus. It's a very big molecule. And when the fungus infects the plant, it pumps a lot of these compounds in there. And that actually self-assembles and makes a pore in the membrane. So that all of a sudden, all of the content of the cell comes out and the cells die. And cercosporium is also cercosporium is very interesting also because it's a photodynamic compound. This is fully conjugated and it's a bright red compound so that the fungus infects the plant. It releases this molecule in the plant, and when light comes on, it becomes photodynamic and it destroys the plant. You know, if you're familiar with how PPO inhibitors work, you inhibit porphyrin synthesis and you get accumulation of porphyrin in the cytoplasm, and that becomes photodynamic and it kills the plant. Well, cercosporium does the same thing. All right, so that's interesting how natural products have evolved to affect membranes. So salmontine is a compound that I'm going to focus on. We published this recently. So salmontine is a compound that's produced by some type of long peppers, the pepper plant. And when you spray it, you get that kind of burn down effect. And this is, I believe, salmontine, and this is pelargonic acid. So on the face of it, it looks like it acts very much like pelargonic acid, that it just washes cuticles off like a soap, like a herbicidal soap, and then the plant dies. But actually, there's more to it than that. We've done some ultrastructure work. That's the surface of a velvet leaf. And if you get closer, when you spray salmontine on top, you can see that it accumulates in the grooves of the plant right here. Now, I'm kind of a geek about this. You know, when you do electron microscopy, you get black and white pictures. But there are tutorials on how you can color your images. So I spent, I don't know, a whole month last year learning how to do this. And I think it's so cool because normally this is all gray. And so it's hard to see the stomata. By putting color, you can tell the difference that actually you enhance the amount of information you get from your images. But anyway, so what happens within the first few hours is the salmontine accumulates in the grooves. But if you wait for 8 hours, you can see that it's absorbed and the cells below it collapse, right? So, and we couldn't see any difference in the cuticle. So we felt like, okay, it's not a herbicidal soap. It actually goes into the tissue and does something physiological in the tissue. And so we spent some time working on this, some very simple assays with electrolyte leakage. And what you can see is that at the higher doses, you basically get very rapid loss of membrane integrity, and that happens in the dark. But some of the lower concentration, when you put the light, you all of a sudden increase the activity. So it looks like there was two processes, one which was light independent and one that was light dependent. And so we wanted to know exactly what happened. So as far as lipid synthesis, which would be the light independent mechanism of action, there are several target sites. Thiolactomycin inhibits that first site right here. Cyprin also inhibits that inulyl reductase. And then there's some compounds like AAL toxin that inhibits this pathway. A lot of work has been done on these compounds, those AAL toxins, because they're very potent and they're produced by a lot of plant pathogens. The problem is these were never developed as herbicides because these were, as my boss Steve Duke says, there have been some studies done on animal toxicology. It makes the animal brain turn into cheese. So you wouldn't want to spray that on something that you're going to eat, right? So what we know about cyprin would be one example that I want to talk about. It's produced by several plant pathogens, and it causes these type of symptoms on the plant, right? Some dose response curves. You can see that you really inhibit root growth. And it actually affects an enzyme called no, this is just growth. So that's a typical dose response with cyprin. And there's an analog called, come on. It's going to be on the next slide, so I'll show it to you right here. Triclosan. See, these two molecules are very similar, and triclosan is a very strong inhibitor of an enzyme involved in fatty acid synthesis. And it inhibits that enzyme inulyl ACP reductase, or ENR, at, what is that, 46 nanomolar. So it's very small concentration. And that's what it looks like on the dose response curve. And when we tested the cyprin, it did the same thing, but at much higher concentration. Anyway, we kind of figured out using computer modeling how that worked. There's some kind of stabilization. This is cyprin right here, and you have the cofactor here, and you end up with what's called pi-pi stacking interactions with the NAD. Those two rings kind of go on top of each other, and they get stabilized. Anyway, so that's another example where nature can do wonderful synthesis for you to discover new ways of killing plants. And there's a lot more. You know, there are inhibitors of hormonal type regulations, and I'll show you one of them. They are inhibitors of adenyl succinate synthase. They are inhibitors of peptide deformylase. Cantharidin inhibits serine threonine protein phosphatase, which really messes up protein expression. And then a lot of histone deacetylase. So I'm going to show you a couple of examples of that we've worked on. So this is really interesting. This is a work that was done by Dow Agro. They isolated this compound from this species, Nectria. It's called cynacidin. And it's kind of, as far as structure, it's a very unusual structure. I'm just going to skip a little bit and I'll show you this. So what's really interesting is if you put side by side the structure of that inhibitor to the structure of jasmonic acid, they're very similar. In fact, if you overlay them, that's what it looks like. So essentially, cynacidin acts like jasmonic acid. It's a hormonal mimic. And I think this is like toward the end of my talk. That's the last project I'm working on. As I mentioned, I was transitioning from the USDA to Colorado State, and that's the one project I'm trying to finish before I leave. So we had some money from a company called Marrone Bioinnovation, the natural product herbicide company in California, and they have identified this molecule MBI 010MR540, long name. But it's produced by this organism right here. And if you put it in the soil, so it's a natural product, you put it in the soil, and over time, it's taken up through the roots, ends up in the foliage, and you end up with necrosis like this. So it's a soil-active natural product, which is quite interesting. And that's what it looks like, okay? It's a very complicated molecule, but it's very similar to this one right here. And the key thing is you have that bridge with a disulfide bridge right here. You have it on the outside right here. And what happens is, well, we worked on it for a good while. We could not figure out what it did until we realized the similarity between these two molecules. And we know that this compound is an inhibitor of histone deacetylase. Now, what do histone deacetylase do? It's very important for gene expression, because in your, most of your DNAs in the chromosome is very condensed, and it's kind of not doing anything. It's very stable and condensed. But if you want to express a gene, you have to unpack the chromosome, and then the DNA is wrapped up around histones. And what happens, so start with the bottom here. So the DNA is very stable in the form of chromatin. When you want to express a gene, you have to unwind the DNA from the histone, and that's done by a set of enzymes called histone acetylase and histone deacetylase. One puts an acetyl group, the other one removes it. And what happens is when you acetylate the histone, you make it more open, and then you can actually end up with DNA replication. Here's what happens. Let me see. So what happens is if you block the deacetylation, so a histone deacetylase removes the acetylation, right? Now, if you block that, then your histone stays acetylated, and this is what you observe here, that this is normal level of acetylation, and as you put an inhibitor, you end up with more acetylation. So essentially, you're stopping the removal of that acetyl group. And that affects all of this. It affects every aspect of plant growth. So the reason why I wanted to finish with this is because natural products not only help you discover novel target sites that are very specific for pathways, but they also take bigger pictures. They actually can develop inhibitors of master regulators. If you inhibit histone deacetylase, you mess up basically all of the plant physiology. And so here's what happens here. With that compound from MBR, 20 macromolar, you get those spots of toxic response. And we showed that you have some level of inhibition of histone deacetylase. Our compound is this one, and this is a natural product that's known to inhibit this. So the activity was not very good. This is one macromolar here. It's pretty high. Now, what we thought about is maybe because the compound has to be bioactivated, and what happens is this disulfide bridge right here, the SS bridge, can open up, and you end up with a free S group. And we did some chemistry where we could convert the oxidized form to the reduced form. So basically, you start with the one with a double bond here, and then slowly you can make the one that's reduced. And what happens is actually you increase the activity of your compound. And that's because the way the histone deacetylase works, you need to have something with a long arm to reach inside to inhibit. And that by reducing that double bond, that bond between the two sulfur, you end up with a more active molecule. So I know it was all over the place because natural products really hit plants all over the place. My main goal is to maybe get you excited about the fact that although we've not had a new target site for herbicide released recently, I know that companies have been looking at natural products. I've been invited to speak to many companies about this. I'm actually going to go to Monsanto this spring. So even Monsanto is interested in natural products. That says something, right? All right. So I want to thank you for your attention. Thank you for the opportunity to be here. Merci beaucoup. Do you have any questions? Yep.
Yes. Do we have any questions?
It's probably just like drinking out of a fire hydrant. Too much information.
Yeah.
So the question is, why are there not more herbicides that are either natural products or derived from natural products? Is that what you're asking? So yes, there's several factors for that. One of them is most natural products have evolved because their mechanism of delivery is through, let's say, a pathogen. So the pathogen infects the plant and releases the compound in the plant. Well, so these compounds then are not optimized for foliar absorption, for example, because the fungus will produce the compound in the plant. So now if you take that natural product and try to make it as a herbicide and spray it over the top, it may have completely the wrong physical chemical properties to act as a herbicide. So that's why, for example, the trichetones, they discovered the target site using leptospiromone, but they had to elaborate the structure so that they'll be more performant as a commercial herbicide. So that's one aspect, is the structure may not be optimized for agricultural use. Second, most of the natural products tend to not hang around in the soil as long as synthetic compounds, or they tend to degrade with light or water, so they're not as stable as some of the synthetic compounds, so that's another factor. The third one would be the cost of synthesis. Most of these natural products are quite complicated. They have what's called chiral centers, which gives them stereospecificity. And so for synthetic chemists, that's really hard to do that. So there's many factors. Other factors would be that, okay, some of them inhibit targets in plants that also have targets in animals, like AAL toxin. It would be a great herbicide, except that it's toxic to everything else, right? So there's many factors why you have to hit that sweet spot of a plant-specific process on a molecule that's not too expensive to make, that sticks around long enough, that can get into the plant when you apply it. So there's a lot of things that have to be taken into consideration. Another question? I think I'm the last person before lunch. Thank you, Linda.
Thank you so much.
Thank you.
Fabulous.